Hello, everyone. We're going to get started on the next big group of fish, keeping in mind that fish goes in quotes, it's paraphyletic. The fish line includes us. And we're actually headed down now, and we're in the point in this class now where we are headed down that line that's going to lead toward us. Making sure I'm recording here. So we've left the chondrichthyes, the cartilaginous fishes, behind. That split goes back to the beginning of vertebrates practically. The first fishes. We left that line behind now, and now we're on the line leading to that leads to the vertebrates we know of as the bony fishes, which includes us. And the ray fin fishes, which we mostly think of when we think of fishes, which are not us. So anyway, there's two major groups here: the Actinopterygii and the Sarcopterygii. The Sarcopterygii includes the tetrapods, us, the amniotes. There's a really nice page here with background information. You don't have a textbook in this class, so information like that's a good background source for reading. Remember, by the way, I think I've said this before, but the little number up here refers to the learning guide question that corresponds to it. What do we know about the osteichthyes, the bony fishes, as far as evolution? Well, mostly from fossils, and there is an excellent fossil record. Why is there a great fossil record? Well, think about a fish that dies and lands on the bottom of some surface, and it gets covered in sediments, or it's a landslide under the ocean that covers and kills everything that it smothers everything down there instantly, and is preserved. Cold, dark waters, not a lot of microbial life to degrade the tissue. And then also freshwater environments as well. Volcanic ash flows that snuff out all the life and compress it underneath layers that become sediment. So we have a great fossil record. Another thing about fish is many of them are fairly flat. So that's going to leave a good fossil, if you think about it, because that's going to fit really nicely between layers of sediments. Where something that's more tubular and round, you have, it's going to be more likely to be broken. It's just more three-dimensional. So we know a lot. There's a lot of fish fossils, and more are found all the time. So let's look at the phylogeny here of them, and zoom in here. And here we have the osteichthyes. We have left behind, again, we've left behind the cartilaginous fishes that diverged long before. The osteichthyes, we have the two. You can see there's a sister clade. You can kind of think of it as the Actinopterygii, and the Sarcopterygii is sister clades. That's not entirely incorrect, I think, to think of it that way. Look over here at the Sarcopterygii. What do you see up here? There's coelacanth, lungfish, and then amniotes, which includes us. But look at the diversity here in living orders of the bony fishes, especially the Actinopterygii here. Look at the fossil record, especially rich in the Mesozoic, and quite sparse down here in the Paleozoic. But I don't know exactly why that is. But if you look up here at these silhouettes, some of them will be familiar, like eel-like fish, gar-like fish, sturgeon-like fish, and then your sort of perch-like fish. Tremendous diversity. I will, in the next video, I will show some pictures from our last trip to Nenegrit Pond. We went out and we saw got some interesting species. So oftentimes you generally define a phylogenetic tree more by the nodes, and the nodes are defined by the structure that evolves. So if you look here, you have the chondrichthyes here, and then at some point there was a split, and on that evolutionary line, we have the evolution at some point along the way of endochondrial bone that we associate most of your bones with. Not talking about dermal bone, endochondral bone. We have the evolution of a lung, which doesn't fossilize very well, or a swim bladder, the homologous. And also the evolution of a bony operculum. Not just a gill opening, not a gill pore, but a bony opening. And you can think about the transition here is toward I say toward because it implies teleology, but it's a transition toward an active, sort of more sophisticated mouth that is in terms of what it can do, the rate at which it can grab something, extend itself, and even support teeth and even secondary jaws. So once we see the evolution of that, and then along the way, you have higher and higher. What this represents is higher up in the fossil record. We have structures and groups that evolve later in time, and we define that clade by things like, is the maxilla mobile or not? Ours is immobile, but that's secondary. The vast majority of the so-called fishes are in the Actinopterygii. They are extremely diverse group. It makes it very hard if you take an ichthyology class. That's the group that's going to, freshwater and saltwater fishes. It's a tremendous amount of diversity. Probably, second only to, you take field botany, and there's a tremendous diversity there as well. But the fishes are very, very diverse. And it's actually a really hard group to learn. Take a look here. You have the Osteichthyes, the Actinopterygii, and we're now going to leave, for temporarily at least, leave behind the Sarcopterygians, where the fin skeleton extends into limbs. So we're going to leave the lobed fin fishes behind for now and come back to them later. Actinopterygii. This is the group we're talking about here. Let's take a look at some of the names. Cladista, not a familiar name to most of us. Acipenseriformes, it should be familiar to you, the sturgeons and paddlefish. Gars and pikes. And bowfin. You have that bowfin skull that I bring out a lot because it really it's a great illustration of dermal bones. And then there's a Lopomorpha, which are the eels. And then there's the teleost, the euteleost. And that's the group there. That's a monophyletic clade as far as anybody knows. All evidence points to that group being a monophyletic clade, which means that everything that's in there is inferred to have a common ancestor. We get back to the common ancestor, we've included all the descendants. We're not arbitrarily leaving anybody out. The diversity. So 45 orders. That's amazing. 435 families, at least 28,000 species, and new species are being found all the time. Imagine deep oceans or parts of the world where there's not been a lot of exploration around the even in shallower waters. How about freshwater systems in tropical environments like up in the Amazon River Basin, in the Congo? That's why new species are found every month of fish. It just keeps adding and adding. Of course, they're not. They were there all along. The diversity in number of species and also in form and in function, where they live, how they eat, how they move around, how they reproduce. There's some species that are of which there are no males. There are some species that can switch between male and female. Species that glow, species that live for a long time. You name it. It's often been said that a child could draw a fanciful picture of a fish that child created in the child's mind, and there probably is a fish somewhere out there that looks kind of like it. They've evolved so many different fin structures. So you don't really talk about diversification into many, many niches. Oftentimes the niches they're evolving, they evolved into here involve mouth parts. So again, remember I said the evolution of the jaw is so important, and not just because it's the thing that once we headed down the line toward jaws, we never lost our jaws. And it fossilizes well. But really, it's also functional. But it has to, because it's so closely linked to how the animal eats. And is it, can you think, when you think of like function and what's important, isn't how the animal eats almost the most important thing you can think of? Of course, how the animal mates, which is another important thing. But how the animal eats, what does it eat? That is, if you were to describe that, you could probably start thinking about what the jaw looks like, because the jaw also forms the facial structure. It houses the teeth. It determines largely what it can eat. So if you look at the sort of the faces of fish, tropical fish, fish you buy, part of the reason people like them is their color. They're also very colorful. That tells you they can actually see in color, which is really interesting. I think of that as a primate trait. But they are, it's also really interesting given that they live way down in the water, where light, not a lot of light. But the shape of the face is really important in the fishes, because the shape of the face reflects the shape of the jaws, upper and lower mandible, and the operculum, and the volume of the mouth, what it eats and how it eats. What you're seeing here are different shapes of jaws and different ways of eating. This also includes different ways of concealing yourself. So if you go to Mystic Aquarium, I believe you can see the garden eels there, which are hilarious. So the eels that suck back down like worms. Of course, some flatfish. We did not catch flatfish this year. Pleuronectoformes, the flounders and halibut. An amazing adaptation there. So this is really a reflection of avoidance of predators. And much of fish growth is really about, a lot of it's about outgrowing your predators, getting large as you can, and having, in some cases, spiny fins. Although many fish don't have spiny fins. Most of them are in the infraclass Teleosti, also known as the teleost fish. Within that group, you have the largest order of vertebrates that there is, which is the perciforms. 40% of all bony fish species, period, are in the perciforms. There's more perciforms than there are living bird species. Tremendous amount of diversity just in the perciforms. This actually causes problems with trying to resolve phylogenies and hypothesize about evolutionary relationships, because there is so much diversity. There's such thing as having too much of a good thing, I guess, in terms of morphological characteristics. What I mean is that there's such diversity. If you look at the phylogeny paper here, see if I can find it on the phylogeny. This is a Tree of Life, new classification of fishes. We have here the Tree of Life of fishes is in a state of flux because we lack a comprehensive phylogeny that includes all major groups. Okay. But a rapid influx of molecular studies is changing many established systematic concepts. So this is a molecular study, which most phylogenies now are molecular studies, where you use markers, where you have to attach primers and you sequence between them. Or you do whole genome sequencing, in which case you still have to select portions. And I don't actually see the tree. It's very complex material. Oftentimes, the problem here is the amount of genomic information is so large that it requires a bioinformatics expert and really complicated statistics to be able to analyze. This is just a picture showing the great morphological variation among the perciforms. Look at that. There's pretty much every shape you can imagine is there. So physical characteristics, I would say there's too much diversity. What happens is when you have many different lines that have sort of the opportunity over time to form a structure, they'll form it. The structure will evolve independently in two different lines. That's convergent evolution. So there's convergent evolution can happen in any two different lines. But when you have lots of different possible pairs of lines, and you have more opportunity for a structure to be shared not through common descent, but through convergence. So that's why there is. And also oftentimes there's a gradient between structure. So it's hard to find the break points where you call something this type in this type. Extremely hard. How there was, you get much less convergence evolution with genetic markers, and there's just more information to draw from. The similarities in genetics is more faithfully tells you about evolutionary relationships than does morphological in some cases. Body size, vast range in body size, including extremely small, like this little fish right here. This is an adult fish, weighs less than a gram. And then you have the mola mola, which is an absolutely giant bony fish here. You can, and the giant, you can actually occasionally see them here in Narragansett Bay and off the. I saw one once off of Jamestown. Couldn't tell what it. We thought it was like an old discarded surfboard. I looked closer, and it was the only mola mola I've ever seen. And then look at the larva, tiny little larva there, mostly eye. Includes fish like this for the oarfish, which people rarely see. They might be more common than we know. A lot of fishes just aren't seen very often. So here we have a magnificent oarfish here. Right underneath that kayak. Large fish, which is very poorly understood. I see that a lot, that so many of these fish species are just, there's not a lot known about them. Very hard to do research in marine environments. The perciforms diversified in the Cenozoic. That should be a kind of a familiar theme. You have the Mesozoic, the Triassic, Jurassic, and Cretaceous. That's the Mesozoic. You think of that as the era of dinosaurs. At the end of the Cretaceous, you have mass extinction from the KT boundary from the massive bolide and probably other volcanic events happening at the same time. You enter the Cenozoic. Cenozoic is when you have the great diversification of flowering plants, of mammals, of birds, and also the perciforms. So all that was happening at the same time. Great extinction event actually opens up lots of opportunities for niches. How anybody survived that is beyond me. Just to give you an idea here for so you don't have to memorize these names in terms of just the names here on this list, but just gives you an idea of some names you should be fairly familiar with, which I've already gone over some of these, and some of these you recognize from lab. But so what are some general characteristics? And we're going to contrast these with chondrichthyes. Well, of course, they're called the bony fishes. In particular, the ray fin fishes. So we fish here the Actinopterygii. We're not talking about the Sarcopterygii. So we're talking about the osteichthyes that are the Actinopterygii, not the Sarcopterygii. Bones, although we have bones as well. So you have, you have the formation of bone, calcification of tissue to form a bone. That did not happen in the sharks, at least not to the extent that you see. The sharks do not have a bony skeleton. They have some bone-like structure in them, but they don't have a bony skeleton. The strength and all the things that bones do for us, they do with mostly cartilage. It's a bony endoskeleton, and once you go down this line, you've developed a bony endoskeleton. We never went back, evolutionarily speaking. In lab, I've had those bones out, those skulls, so you can see the different characteristics and find those bones. One thing I oftentimes have two different fish types of fish we use to illustrate this. There's the bowfin, which has a sort of a shield of dermal bones, and then there's a couple of mounts of like a smallmouth bass or something like that, a Helios like that. But what you're seeing here is one thing that's familiar to us actually is the large. You have large dermal bones that cover the head. And in the case of fish, the pectoral region. But think about, contrast us with with a bony fish. Is we have a separate head, neck, and the rest of the body. Okay, so there's not a strong connection between the bones of our cranium and our neck and the rest of our body. Those are separate. In the case of fishes, there's a much tighter connection between those and no visible neck. They don't have a neck. They have teeth on their premaxilla, where your incisors are. Premaxilla, or your top incisors are. They have a maxilla, most of your upper jaw, and on the dentary bones. So when we think about dentary bones, we think of our lower jaw, because in mammals, that's the only bone we have left in our lower jaw. We think of premaxilla, we think of our upper incisors. When we think of maxilla, we think of our upper jaw, but our upper jaw is connected to the cranium. It doesn't seem like a separate thing, but it actually is developmentally. Otherwise, the bones, many of these are familiar. You would see these in any mammal bone or that of a crocodilian. Some of them are not, like the operculum bone. And some bones have evolved into other bones, like the hyomandibula has evolved into a different bone than us. So it's pretty amazing, frontal bone, parietal bone. So at some point, you have the evolution of these dermal bones, the basic structure of a cranium, and that is what was passed down to all the descendants, and that's what we all have at that point. So pretty obvious here what's going on. We have the evolution of a bony housing for the head, protecting the head. Also, any kind of bony structure oftentimes is a place for muscle attachment. Just compare some more cranial bones here. We have our upper jaw, which is the maxilla, and it's fused with our cranium, even shaped here to form our orbit and zygomatic arch. That is not whereas that's not really the case for this bowfin here. We don't have an operculum bone anymore, but we do have a dentary bone. The fish does have, but the fish also has an angular bone and a serangular bone that contribute to the lower jaw. In mammals, those bones do not function in the lower jaw anymore. They're somewhere else, but they're not there. Operculum. Now, this is a really key adaptation. In this video here, I think we can see that here. Here are some fish breathing. I just use this video just because it does show operculum function. Not very well, though. Not all fish do this, by the way, and even goldfish don't always do this. They don't have to really do this to breathe. It's not really clear what they're doing here. It's clear to somebody, but notice here, look at this. Look, you have the motion of the mouth opening and the operculum opening. They're coordinated. When one closes, the other one opens. Close, close, open, open. It does that to control and regulate the flow of water and materials in and out. Because the problem with having an operculum is that food that enters the buccal cavity could escape out the, and you know that if you ever catch fish and put them on a stringer. Right? And also to create suction requires shutting off and opening the operculum like a valve. So it prevents backflow and regulates buccal pressure. So think about suction. You see some examples of that. It does power the ventilation so that gills are the most efficient respiratory organ in all of vertebrate biology. However, it only works well when there is a more or less constant flow of water moving across, because those gill filaments will take out the oxygen out of the water that it's in contact with, leaving it with relatively unoxygenated water. So you want to get that passed out through, replaced with water that has oxygen. You constantly have, if the water didn't move, you had depleted water, oxygen depleted water right against the gills. On the other hand, this is where they give up CO2 to the environment. So same thing, you would have a high CO2 environment right there around the gills, which wouldn't be a good thing, trying to get rid of CO2. They're extremely efficient. They're also very sensitive. So you know if you ever catch fish, these gills can bleed. They're very sensitive. Once you catch fish and manipulate their gills, there's a good chance they're going to die or be injured, not live as long. Gotta be really, when you throw back a fish, gotta be really careful. Catch and release is great, but actually, obviously, it doesn't do the individual any good at all. There's a really nice video here talking about the way swim bladders work. One of the problems, of course, fish have, it's on the good side, is they have equal pressure all around them, and they don't have to worry too much about gravity. They will sink. Of course, there is some gravity, but mainly the pressure is all around. So they don't have to really push off of anything except for water in all directions. But as you know, when you walk through water, how hard that is. So water, pushing against water is actually quite difficult. These fish have very strong muscles. But the problem they have is how much energy does it take to move around the water column? To scoot yourself forward, in some cases backwards, but to get up and then down. So what if you had to use your fins in your body, creating a wave in your body to get up and down? That's an expensive way to do that. What if instead you were like a submarine, modern submarine, or even like a U-boat or something like that from the First or Second World War, where you can rise. Obviously, you think about, a submarine doesn't flap anything, right? They just go up. They go down because they use compressed air. They change their buoyancy. So they have compressed air tanks on the submarine, and they use those to adjust their location in the water, position in the water. And it's a function of Boyle's law, which is nicely described here. There's a nice little video on how that works. They change the pressure inside their gas bladder by secreting air molecules and increasing the pressure or decreasing the pressure that way. So imagine air molecules come out of solution and into gas form, into a gas chamber, gassy chamber, expanding, and imagine that contracting and those molecules going back into fluids. That's going to change your buoyancy. How about sharks? How do they do it? They don't have a swim bladder. And by the way, the swim bladder is homologous to our lungs, apparently. With no swim bladder, how do they do it? Well, they have to swim. They have to swim up and swim down. So they use these paired fins to sort of move themselves, create waves, push themselves against the water. They don't really fly into the water, but they push themselves. But mainly, they're just buoyant anyway, because they have to start with. Sharks sort of have the opposite problem of trying to. It's harder for them to go down because they already are buoyant because of their the density of their inner flesh, and that partly is a function of their large oily liver. Large oily liver and a low density skeleton as well. It's another advantage to the cartilaginous skeleton is they can more easily rise to the surface. And think about how a shark feeds. Oftentimes what they're eating is up there on the surface. They have to go down though is a problem. What else about we're looking at general characteristics that distinguish them. So we have a swim bladder. Also, now we're going to move into something that's, thinking about things that are homologous to the limbs. The first vertebrates that crawled out on land. What do they have? They had fins. They had fin-like structures. At the tip of the fins, what's there? Well, these are structures that fossilize fairly well because they're bony. And what they have is a structure called lepidotria. And they are segmented bones here, and they are called bilaterally paired. So they basically just mean they're paired in the fin rays. So rays go out, they're paired. You have these bony segments. This is really important because if you compare across all the bony fishes, you can see the similarities and get a hint about what the common ancestor looked like in terms of its limbs. So at some point, you have a bony fish that evolves limbs. What does it have to work with? We're talking about the Actinopterygiae. Here's what they have. They have lepidotria. Look close up here, and you can see those lepidotria. You see how they, at the end, they fan out and form these bilateral rays. Connected with tissues, this is fairly familiar. It's kind of like webbing, but it's not homologous. I don't think it is. It might be homologous with our webbing, mammalian webbing, bird webbing, but webbing is something you see, you see a lot of that actually in the vertebrate evolution of the vertebrates. What does it do? Well, think about your position in the water. Think about pitch and roll. If you've ever been on a ship, a really unstable ship, you know how frustrating that can be. If somebody gets seasick, it can make you seasick. I was once on a sort of a catamaran kind of like ferry going from Santa Barbara out to Catalina Island. And it was right around Christmas Eve. It was the last boat. It probably shouldn't have gone. And it was just a vomit fest on that thing. Up it went, but it didn't just go up and down. Every possible movement you could describe of that ferry happened. So pitch, roll, yaw, all of it. It did it all. I remember looking out and seeing Venus off on the horizon, and Venus was doing this terrible, terrible trip. No amount of Dramamine could deal with that. We think of fish, we think of scales. Scales are vertebrates. First, what do you come in contact with? Very oftentimes, remember, vertebrates are fast moving. They're oftentimes aggressive in the way they go after food. Sometimes the way they catch their food is they hit it hard, right? So you've got to have a tough skin. Also, you get bitten into. Many fish survive. If you're an old fish, you have survived being bitten by other vertebrates. Well, you're covered in scales. So armor is a really common, we saw this with other fishes. It's not armor, it's not homologous to scales, but armoring, wearing armor, there's good and bad to that, but we see it. And so the nature of the scale, you tend to have 4 different types when you look at the fishes, but there's other types as well. These are the 4 major ones you're seeing here. But something to keep in mind is think about the evolution of scales. Think about the evolution of feathers, evolution of hair. Those are scale. Where do they evolve from? They evolve from scales. Where do these scales evolve from? They evolve from scales, some other scale. Scales are an ancestral. You look at the common ancestor of all of us, that Kenopterygidae and the Sarcopterygidae, they had scales. From those basic scales came all these scales you see here, and hair and feathers. Scales fossilize really well. What's great about them is they're lost. They're non-living tissue. They fall to the bottom. So we got trillions of scales out there as far as fossils. If you go through one of those you pick fossil kind of places, you can dig for your own fossils. Oftentimes that's what you find. I find them in Nevada, some of the limestone ranges of a lot of limestone. Here are the 4 types. Again, in lab, unfortunately, under a scope, it's not obvious because they're overlapping. You can't see the whole structure, but the ctenoid, associated with most bony fishes. Cycloid, which is associated with the salmon. So I think a salmon is almost, some fish, by the way, don't have scales like the catfish, but salmon, the scales are very soft. You can even eat the skin if you want on a trout or a salmon. Cycloid scales, it's because of the nature of the scales, not nearly as, they're much more, less armored. And then ganoid scales, which are definitely harder. A gar is a sort of a semi-armored fish. And I've seen some enormous gar in Louisiana, like that people catch there to eat. Gar is delicious. The Cajun people catch them, but some of them are 7 or 8 feet long, and they seem to make no big deal of catching something like that. It always kind of makes me wonder how old those fish are. But anyway, they're you touch them, they're hard. And then placoid scales, which we discussed, which are found in sharks. Okay, so I'm going to stop there, and then the next slide, next video, we'll look at reproductive characteristics. These are morphological characteristics.